Good afternoon everyone, I'm Joshua Griffin and I'm a medical scientist down in Melbourne at St Vincent's Hospital. I'm here today to talk to you on behalf of R-Biopharm into their new Dermatophytes A 12-well multiplex PCR assay, which promises to distinguish Trichophyton indotineae from Trichophyton mentagrophytes. So the talk will be in two parts, with the first part being a brief overview on Dermatophytes, the common species in this complex and the diseases they cause, whereas part 2 will be about the validation into the new Dermatophytes 12-well A assay itself. So, what's a dermatophyte? Dermatophytes are pathogenic filamentous fungi that can commonly cause skin, hair and nail infections in both humans and animals. They are able to obtain their nutrients via the digestion of keratin, a protein that is found in nails, skin and hair. Infections by dermatophytes are commonly referred to as tinea or ringworm, and the disease that you can see below in those multiple body sites is due to a host reaction to the cell wall of the fungus while it's digesting its keratin. Generally around the world, 20 to 25% of the human population will have a dermatophyte infection each year. Here we have a brief overview on some of the more common species of dermatophytes that you will come across. So we have Trichophyton species, which for today's talk is the most important species that we will go into. You then have Epidermophyton flocosum there in the middle, Microsporum species off to the right, and down the bottom in the left, Nunazia species. So all 4 of these species are currently available for detection on the R-Biopharm Dermatophytes and other fungi 12-well assay. Down the bottom there on the right we have Multiplex species as well. I've just included it for a brief overview as it is another dermatophyte that can cause infection in humans via transmission from chickens and other fowl. So Trichophyton species is a genus of filamentous fungus that can contain up to 16 different species. This number of species has been whittled down from 22 species after DNA sequencing has shown that some species are more closely related to other dermatophyte species or require their own genus altogether. They can be both zoophilic and anthropophilic in nature, either zoophilic being they have animal host reservoirs and then anthropophilic having human host reservoirs. Trichophyton species are characterized generally by their morphological development of microconidia and macroconidia. However, in just a sec you will see that is not a 100% golden rule. And some of the more common species that we can come across within the Trichophyton species genus are Trichophyton rubrum, mentagrophytes, interdigitale, pseudonancy, tonsurans, and violaceum. However, there is a new species on the scene that has emerged within the last decade or so, with it being Trichophyton indotineae, that has an increased prevalence around the world. So here we have just a brief overview of some of the common Trichophyton species that produce microconidia. So at the top there you have Trichophyton rubrum, which is characterized by the production of those clavate teardrop-shaped microconidia. They also have a downy white colony morphology on agar with a bright red reverse color. In the center there we have Trichophyton mentagrophytes, but I'll go into that into a bit more detail in just a second, as it is of quite measurable importance to today's talk. And then down the bottom we have Trichophyton tonsurans. And here we have 2 species of Trichophyton that do not produce microconidia. So at the top there you have Trichophyton pseudonancy, which has a high prevalence in African populations, and then Trichophyton violaceum down the bottom, which can cause a high degree of tinea captis infections. So Trichophyton mentag is the secondly most commonly isolated Trichophyton species in humans, just behind Trichophyton rubrum. Again, it's found worldwide and can cause nail, skin and hair infections. Most human infections are commonly acquired via some sort of animal transmission, so generally your domesticated pets or farmyard animals. However, there has been an increase in human-to-human infections within the last decade. So going into more detail on the colony morphology of Trichophyton mentag, you can see that it has a powdery white coloured colony morphology on agar with a slight yellowy to red reverse depending on the medium of agar you're using. Down the microscope you can see it also produces those clavate microconidia. However, it does produce an abundance of round microconidia as well. It also can be characterized by those rather large cigar-shaped septated macroconidia, as well as some older cultures producing those spaghetti-like spiral hyphae. So, Trichophyton indotineae, the most important species of Trichophyton we'll go into detail today, and the whole reason for this new assay being created. So Trichophyton indotineae is a highly infectious drug-resistant fungus that can cause extensive tinea infections that are very difficult to treat. It has emerged from India within the last decade and has now spread globally, including to here in Australia. Trichophyton indotineae is suspected to have originated due to the overuse of topical antifungal combinations that are readily available over the counter in other parts of the world. In 2020, it was proposed to be a new species from Trichophyton mentagrophytes variant 8 after its internal transcribal region of ribosomal DNA was sequenced and was found to be unique. Generally, Trichophyton indotineae infections respond poorly to treatment, with its resistance to terbinafine, the first drug of choice for dermatophyte infections, being the cause. Infections result in widespread lesions affecting multiple body sites that are easily spread from person to person. That point of resistance in Trichophyton indotineae comes in two different forms. So generally you have, which is the most seen version of resistance, being the terbinafine resistance. This occurs through point mutations in the sequinoline epoxidase gene, or SQLE. This prevents the binding of terbinafine to sequinoline epoxidase due to the changes in enzyme structure and its active site. There's also worryingly an increased resistance to azoles. This can generally occur via the overexpression of the CYP51 gene that causes an overproduction of the enzyme that azoles target, leading to decreased efficiency. And here we have just a brief diagram of how this resistant mechanism to terbinafine and azoles in dermatophytes works. So you can see that those 14 different SQLE mutations can cause the sequinoline epoxidase to change its structure and result in terbinafine not being able to bind to it. Also at the top there you can see how overexpression of the CYP51 gene can lead to azoles being unable to bind to the enzymes that help target the destruction of the fungal cell. So like I mentioned earlier, Trichophyton indotineae has prolificated widely from India within the last decade. It has now reached epidemic levels there and also in Iran. Roughly 78% of all dermatophyte infections in India are now known to be caused by Trichophyton indotineae. From this image you can also see how quickly Trichophyton indotineae has been able to spread across the globe and is now present in all major continents, with the highest prevalence being, like I said, in India and in Southeast Asia. Here in Australia we had our first published case of Trichophyton indotineae in 2024, however it's believed to have been here long before then. Across Europe it is believed that the first published cases were presented in the late 2010s to early 2020s, depending on the country, with first cases in the US and Canada being in 2023 and 2024. However, as you can see, it is still present in South America, parts of Africa, the Arabic countries, and Southeast Asia and Japan. So the colony morphology of Trichophyton indotineae is incredibly close to that of Trichophyton mentagrophytes. You can see that it still has that suede, powdery colony morphology on agar with a yellowy reverse that diffuses throughout the bottom of the agar plates. In the lab we have some cultures where it does even go a more yellowy red color than what is described on this picture. Microscopically, they still have those cigar-shaped macroconidia, however they can be slightly more pointed than the ones you see in Trichophyton mentagrophytes, and they still produce those clavate-shaped microconidia and some little round microconidia. So what does a Trichophyton indotineae infection look like in a person? If you've had a bit to eat before and don't like to be squeamish, look away now. You can see it causes a very painful, itchy, spready rash that can be present on multiple body sites. The rash itself can be crusty and bloody, with the formation of lesions that are particularly found in the groin, buttocks and thighs. However, you can see that those spots of the body are not the only places it can be found. You can see that many patients here have it across their shoulders and back, down their arms, down their legs, their abdomen and even their neck. So with that brief summary of the Trichophyton species, genus, and Trichophyton mentag and indotineae in particular, we'll now go into the validation of the Dermatophytes A assay itself. So why do we need this new assay? As you've already seen, Trichophyton indotineae is morphologically very similar to Trichophyton mentagrophytes. You would not be able to reliably call a Trichophyton indotineae isolated in culture Trichophyton indotineae from its morphological characteristics alone. The current assay available cannot differentiate Trichophyton indotineae from Trichophyton mentagrophytes either, resulting in DNA sequencing being the current gold standard for Trichophyton indotineae infections. However, this means it is more expensive and has a longer turnaround time than a commercial PCR assay. There's a very large public health concern around Trichophyton indotineae due to that tipidin resistance and even its increased azole resistance. We currently don't know the level of Trichophyton indotineae within the community, and the lack of education within the medical community has helped Trichophyton indotineae proliferate around the globe, with it presenting as a similar form of dermatophyte infection or other skin disorders. So we'll just have a brief overview on R-Biopharm's assays and how they work. So the assays use a patent multiplex tandem PCR or MT-PCR that allows for the detection of multiple gene targets in the one sample without compromising specificity or sensitivity. This allows for the lowest quantity of nucleic acids to be amplified, even if they are mixed in higher concentrations of other genetic materials. You can use as little as 1 microliter of sample, allowing you to have more sample to be used on other tests. The technology uses nested primers, which are used to ensure only the target nucleic acids are amplified for detection within the assays. Each target is amplified independently, which eliminates competition between these targets. And here we just have a brief video of how this technology works. So sit back, relax, and enjoy the show.
Multiplex tandem polymerase chain reaction, or MT-PCR, is a patented molecular in vitro diagnostic method used to simultaneously detect up to 40 gene targets associated with medical conditions. After the collection of a sample, nucleic acids are purified and ready for MT-PCR. A two-step nested PCR process. MT-PCR is performed on the high-plex or ultraplex systems by Oz Diagnostics. In step 1, tubes containing outer primer pairs specific to the gene targets offered in the product panel bind to the target DNA or RNA within the sample. RNA targets undergo reverse transcription to create cDNA. Bound DNA strands undergo 15 to 18 cycles of PCR. This low number of cycles minimizes the competition between low concentration and high concentration targets, thereby preserving relative quantitative results and increasing sensitivity. PCR products from step 1 are diluted. This removes potential inhibitory and cross-reacting substances. Each diluted sample is aliquoted across multiple wells of the Step 2 plate. Step 2 of MT-PCR is performed in the MT analyzer. Each Step 2 well contains the specific inner primer pair for every target offered in the panel. This ensures optimal specificity. The inner primers are designed to be nested inside Step 1 outer primers, so only target DNA will be amplified. The dye present in the Step 2 reaction emits fluorescence proportional to the amount of double-stranded DNA produced during the 30 cycles of real-time PCR amplification for Step 2. Presence of a target gene is represented by the fluorescence detected in the corresponding Step 2 well. These results are also presented as melt curves, and gene targets detected in the sample are automatically called for clear diagnosis. MT-PCR allows comprehensive molecular testing. It also allows the easy combination of different targets to create custom panels for the future needs of in vitro diagnostics.
So with that background into how R-Biopharm's range of assays work, what was the aim of the study? The aim was to validate the new R-Biopharm Dermatophytes A 12-well assay for its detection, correct determination of Trichophyton indotineae from Trichophyton mentacrophytes. So currently the Dermatophytes and other 12-well fungi assay is on the market, and this is the one we use within our lab down in Melbourne. So it has all those common dermatophytes seen earlier on the assay as gene targets. So you have your Trichophyton species, your rubrum complex, Menteo complex, Microsporum species, Epidermophytum flucosum, and Nanazea gypsia. It also contains some common yeasts and molds, with them being Candida albicans, Parapelopsis, Galbrata, Aspergillus species, and Scopulaeopsis species, which are two common molds that can cause dermatophyte-like infections in humans. The new Dermatophytes 12-well assay has removed a couple of those gene targets, mainly some of the Candida species and the Aspergillus species, and in their place have been replaced with Trichophyton indotineae and the Trichophyton indotineae reference gene. This reference gene allows for the correct calling of a positive Trichophyton indotineae from Trichophyton mentacrophytes. So all the samples that we used in this validation were either nail, skin, or hair. We started the validation using some stored frozen samples that we had been collecting over the past 6 months. Those frozen samples were frozen lysis buffer inoculations, not the actual frozen DNA extracts. As the validation went on, we used some fresh samples that were coming through our laboratory. So to go into the method and equipment that we used and undertook for this validation, samples were placed in a lysis buffer inoculum, which in this case is the PrepYX tissue digestion buffer from R-Biopharm. After inoculation, these lysis buffer inoculums were heated for 30 minutes at 70 degrees Celsius. From here, 200 microliters of sample was pipetted into a 96-well cartridge and run on our DNA extractor, which in this case was the Roche MagnaPure. This step takes roughly 1 hour. The sample elution from the DNA extractor is then taken and placed on either the high-plex or ultraplex systems by R-Biopharm. This is where the step 1 PCR reaction that you saw in the video earlier occurs. This takes roughly 2 hours on a full run of 96 samples on the ultraplex, but can be a little faster with smaller sample sizes or if you were running it on the high-plex system. The assay plate from the step 1 PCR reaction is then moved on to the MT analyzer for the step 2 PCR reaction, as well as the reading of those fluorescent gene target wells to produce the run results. This step again takes roughly an hour. So all up, for running a run of samples for this validation, the whole process can take around 5 to 6 hours. Just a brief picture of the machinery and buffer that we used for the validation. So at the top there in the left-hand corner, you have R-Biopharm's PrepY DX digestion buffer, which works amazing for digesting nail and skin samples and releasing that DNA that we need. We then have the Roche MagnaPure there in the top left. And then down the bottom we have our high-plex and ultraplex 3 systems by R-Biopharm. So the high-plexes can run 24 samples, or in our case we were running 22 samples plus a positive and a negative sample. And then on the ultraplex we were running 94 samples plus again a positive and negative control. So throughout the validation, 508 samples were conducted and run in parallel on the existing Dermatophytes and other fungi 12-well multiplex PCR assay and the Dermatophyte A multiplex PCR assay. Of 472 of these 508 samples, they were all clinical in nature, either being nail, skin or hair, like I said earlier, whilst 36 of them were direct fungal cultures. 30 of those direct fungal cultures were known Trichophyton indotineae isolates that were provided graciously by the National Mycology Reference Centre over in South Australia. So just a brief overview on the types of samples that we use. So 70% of all the samples within the validation were nail samples of some description, so either toenail, fingernail, or nail clippings that had no site stated on their point of entry within the laboratory. Roughly 25% of samples were skin in nature, so with most of them being either leg, groin, or foot scrapings. And then we had about 2.5% that were hair or scalp samples. I've just pulled those two types of samples together as a lot of the scalp samples had contained hair within the sample inoculum. So, what did we find when we ran this new Dermatophytes A 12-well assay? So, roughly 245 of the samples were positive for Trichophyton rubrum. This generally isn't very surprising, given that the most common dermatophyte that causes infections in humans across the globe is Trichophyton rubrum. We had 150 negative samples, followed by roughly just over 70 samples being positive for Trichophyton mentogrophytes. We also had 33 samples positive for Trichophyton indotineae, which was a great result. And then we had smaller numbers of positives for the other gene targets. Comparing these results between Dermatophytes and other fungi 12-well assay and the Dermatophytes A 12-well assay, we can see that mostly all the samples had their gene targets match up between the two assays. There was a slight drop-off on the Trichophyton mentog positives between the two assays, as some of those samples were actually called Trichophyton indotineae in the new assay. I'll talk more about this in just a sec. A couple of those Trichophyton mentogrophytes, however, with some Trichophyton rubrums and one Epidermophyton flocosum, had their gene targets drop off in the new Dermatophytes A 12-well assay. We're hypothesizing that this may have occurred due to the freeze-thaw process that we undertook, as this was generally seen in some of the older samples that had been frozen and run in the earlier parts of the validation, as we didn't see any of this with the fresh samples towards the end of the validation with those fresh samples. So all in all, we had about 97% concordance with only 3% concordance, which again, we're hypothesizing that this was due to that freeze-thaw progress in those earlier samples run in the validation. I mentioned earlier that more on those Trichophyton and Mentagrophytes samples that were called Trichophyton and indotineae in just a minute. So this is what I was generally referring to. For some background information, our lab at St. Vincent's, Melbourne, does not routinely do culture for dermatophytes anymore. We are all entirely using the current dermatophytes and other fungi 12-well assay. However, there are a couple of notabilities when we do culture, and this is one of those cases. So when we have a request slip come into the laboratory that is querying a resistant fungal infection or Trichophyton indotineae, we set up a culture on our derm agar medium with a urea slope medium. While this culture is going, we await PCR results. So when those PCR results come back and they are positive for Trichophyton Mentagrophytes, and we grow a sample fungus that resembles Trichophyton Mentagrophytes, you'll remember that I said earlier that Trichophyton mentag and Trichophyton indotineae are indistinguishable from one another in culture. We will then look at the urea sample, and if it is negative after 7 days, we will send that Trichophyton mentag complex out to South Australia to the reference laboratory for sequencing. So while we waited for those sample results to come back, we ran our samples through the new Dermatophyte A 12-well assay, and they actually all came back as Trichophyton indotineae. So across the validation, we had 3 of these such cases come up, and all 3 of them were Trichophyton indotineae on the new Dermatophytes A 12-well assay. So we couldn't actually report these results out at the time, as this assay is currently not TGA approved, and yeah, we just kind of had to wait for those important sequencing results, which eventually came back as the sample being Trichophyton indotineae. So in conclusion, what can we draw from this validation? The study has demonstrated that the new R-Biopharm Dermatophyte A 12-well assay is a reliable method for diagnosing Trichophyton or other dermatophyte infections in skin and nail samples. The assay can reliably distinguish Trichophyton indotineae from Trichophyton Mentagrophytes. The assay itself will allow for a faster diagnosis of Trichophyton indotineae without labs needing to send out samples to reference laboratories for costly and longer turnaround time causing sequencing analysis. This again will cause faster treatment options and patient care outcomes, whilst lowering work impact on laboratories, being either hospital grade labs or reference laboratories themselves. The assay will also allow us to substantially monitor the levels of Trichophyton indotineae within the population that are currently being overlooked or missed, as you can see from those samples that we received during the course of the validation, that even though some of them were querying a resistant fungal infection, they weren't getting called Trichophyton indotineae on the current Dermatophytes and other fungi 12 assay, but were being called Trichophyton and Dermatophytes. I'd like to acknowledge Sarah Kidd and her team over at the National Mycology Reference Centre for supplying those Trichophyton indotineae cultures, as well as doing that sequencing for us. To my boss, Dr. Jaron Dardine, and the rest of the team at St. Vincent's Hospital in Melbourne for their support whilst undergoing this validation. And to R-Biopharm for allowing us to undertake the assay validation and sponsored me to come up here to the NRL conference and share all our findings today. Thank you.